So welcome to this third lecture in the series. In the last lecture we saw the force field, which allows us to calculate the energy of a molecule as a sum of terms. Now we can actually start to use it for various things. The first thing is to calculate the energy for potentially any conformation of the molecule. We can take the molecule, the coordinates of all the atoms. We can then work out all the bond lengths, all the bond angles and so on. We can feed that information into a force field and calculate the potential energy V. This energy is a function of all the coordinates in the molecules. So we've got the coordinates of atom 1, x1, y1, z1, the coordinates of atom 2, up to the coordinates of atom n. So therefore this energy is a function of 3n coordinates. So we've got q here, which is just a generic coordinate to the power 3n. We've got 3 coordinates for each of the n atoms. If we write q, our coordinates as a vector, so each vector component q has 3 components, we can write it as bold q to the power n. This is because conformational space has 3n dimensions, where n is the number of atoms in the system. Now we can calculate the energy for a given conformation of the molecule. We can start to explore conformational space. So the first thing we would like to do when we have a molecule is to perform a so-called energy minimization. That's really a question of, is our structure stable? If we had this structure and we let it go, would it stay where it is or would it change? If we're at a minimum on a potential energy surface, the gradient of the energy is equal to 0. It's a flat point on our energy surface. So if we're not at a minimum and we let it go, it would go downhill until it reaches a minimum on the surface. Mathematically, we can characterize a stationary point or a minimum on the surface by saying that the derivative of the energy with respect to all the coordinates is equal to 0. So it's a flat point on the surface in all directions. If we're not at a minimum, then we can work out which way is downhill by working out the derivative of the energy with respect to all the coordinates, and then taking the minus sign of it, so it's the negative of the gradient, and this gives us the downhill direction g. And then we can take a step in this downhill direction and change the coordinates of all the atoms so that we move to a point with lower energy. So in this example here, if we started off at this starting point, we can find that the gradient is not equal to 0. So we're not at a stationary point. We can now work out that the gradient points this way, so we can take a step down the hill and we can keep going and take steps until we get to this minimum on the surface here, where it can't directly go any lower in energy. It's interesting to note on this plot here, if we started over here it would take us down here, but if we started on the other side of this peak, it would actually take us down to this stationary point here, this minimum. So there are different ways of finding a minimum. There are approaches where you don't need to calculate the gradient. Here you just basically do a grid search, so you divide your conformational space into a grid and you do a calculation on all the points, and eventually you'll find the one which is lowest in energy. That's not a very efficient way of exploring conformational space, but it works for very small systems. A more sophisticated way is an approach which uses the first derivative. So this is very similar to what I just described on the previous slide. So you're at a point, you calculate the gradient of the energy and then work out the downhill direction and take a step. There are various approaches which use gradients, which you'll come across in the literature. The first is so-called steepest descent, which basically, if you're at a point, say here, you find out which is the steepest direction downhill and you take a step. And then when you're at the next point, you find out which is the steepest direction downhill and you take a step. There are slightly more sophisticated methods which exist, which use a history of steps. So it remembers the type of step that it did before, and this often can find a minimum more quickly. And one of these approaches is so-called conjugate gradient. You'll see these in the practical classes. There are also some more sophisticated ways of finding a minimum, which involve the second derivatives of the energy with respect to all the coordinates. The matrix of second derivatives is called the Hessian matrix, and we'll revisit this later on in this lecture. And one of these examples is the so-called Newton methods. So ABNR stands for adoptive basis Newton-Raphson. So this is one minimization technique which uses second derivatives in order to find a minimum. And the idea is that making these methods more sophisticated means it's faster to find a minimum, or it requires fewer steps, or that sort of thing. Once you've found a stationary point, so a point where the gradient is 0, then you need to be able to classify it. So there are different types of stationary points you can get on a potential energy surface. So here is an example of a potential energy surface where you have an angle and you have a distance. And you can see here you've got a minimum here, you've got a deep minimum here and a minimum here, and this point here is a transition state. So if you think about it, the gradient at the minimum and in the transition state are going to be 0, but they're very different types of points. So if you've got a minimum, then the first derivatives are equal to 0, and all the second derivatives are positive, and that tells us that we're at a minimum. So this is the case for points like this at the bottom of a potential energy surface. You can also have the gradient equal to 0 at a first order saddle point. So this is an example here. This is a transition state, and at this point you will have the gradient in all directions is equal to 0, but the second derivatives, if you're at a first order saddle point, then one of the second derivatives will be negative, but all the others will be positive. So what you need to do to work out whether you're at a minimum or not is calculate all the second derivatives and check whether any of them are negative. And if they are, then you're at a first order saddle point, or perhaps at a higher order saddle point. If you've got more than one second derivative which is negative, then you're at a higher order saddle point. So here we have one coordinate which is one second order derivative is negative, and here's a second order derivative which is negative. For chemistry and for biophysics, first order saddle points are of interest, but second order and higher order saddle points aren't of any particular interest. So if we're interested in saddle points, transition states, these are first order saddle points. Now, you'll have seen in that plot a few slides ago where we had a curve, potential energy curve, that you can have global and local minima. It's actually quite easy to find a minimum. You just start from your conformation and go downhill, for example using a steepest descent or a steepest descent algorithm, for example. But when you're at a minimum, you don't know whether you're at just a local minimum or at the global minimum. In fact, it's quite difficult to find the global minimum. If you think about it, one way would be to find all the minima on the surface and then work out which is the lowest energy. But if you're in a very, very large conformational space, like for a protein, this is very, very challenging. This is actually an active research field, how to find a global minimum. There's a web page here you can have a look at, and this is a plot which illustrates some work coming out of Professor Wales's group, and they're looking at the energy landscape of a short peptide. So there's two conformations. Here is the alpha helix, which has a lower energy down here, and this area in blue is a beta sheet type conformation. And this diagram here, we don't need to worry about the details, but it shows that if you're in a beta sheet conformation, it's a local minimum, whereas it's the alpha helical conformation which is the global minimum. But it also shows you that if you want to go from the alpha helix to the beta sheet, then you've got to go all the way up here. So there's a huge energy barrier. So this is energy up here. There's a huge energy barrier in going from the alpha helix to the beta sheet, or vice versa. And this is interesting because it shows that if you go into one minimum, a local minimum, you can actually get stuck in a local minimum, and you might not even be aware of the global minimum being elsewhere on the energy surface. So let's say you found a minimum on the surface, either a local minimum or a global minimum. What can you do when you get there? Well, the first thing you can do is something called normal mode analysis. And this looks at the vibrations of the molecule at that point on the surface. So a normal mode you'll have come across already. They're the vibrations that you get, for example, in infrared spectroscopy. So here's a definition. So a normal mode is an independent synchronous motion of atoms or group of atoms that may be excited without exciting other modes. So they're completely independent of each other. So if you take a molecule like CO2, there's the symmetric stretch and the asymmetric stretch of the molecule. And these are completely separate, and you can excite one and without exciting the other. Why might you want to calculate normal modes for biomolecules? Oh, so I've mentioned these are the vibrations relevant to infrared spectroscopy. When you're thinking about vibrations in the molecule, you'll remember the equation 3n-6. So for nonlinear species, and most biomolecules are nonlinear, there are 3n normal modes in this system of n atoms. So why might you want to calculate these for biomolecules? Well, low frequency normal modes are typically delocalized and describe large scale movements of the system. And often these modes are associated with function. It could be, for example, the opening and closing of an active site in an enzyme, or it could be a protein which switches between two conformations, an on conformation and an off conformation. So one example is with the protein HIV-1 protease, which is this dimeric protein here, and here you can see the structure with an inhibitor bound to it. Above the inhibitor pocket there are these two flaps, and these two flaps actually open and close to allow access to this binding site. And the motion of opening and closing of these flaps is actually one of the low energy normal modes of this system. So here's an animation which I've taken from a website which illustrates the normal mode for one of the monomers in HIV-1 protease. So you can see this flap is moving from one configuration to another configuration, and this is actually opening and closing the active site of the enzyme. So these normal modes can give insight into the function of various proteins. So how do you go about calculating normal modes? The first thing you need to do is find a minimum. If you're not at a minimum, the calculation of normal modes doesn't really make any physical sense. Once you're at a minimum, the point where the gradient is equal to 0, so all the first derivatives are equal to 0, you then need to calculate the matrix of second derivatives. As I mentioned before, it's called the Hessian matrix. Once you've got the Hessian matrix, you need to make it diagonal, and that gives you a matrix with numbers along the diagonal, but 0 elsewhere. When you do this, it gives you eigenvectors, which are vectors describing the motion associated with the normal modes. And you also get eigenvalues, the vibrational frequencies associated with these normal modes. This is just a mathematical task. You can do it by hand, but normally you'll just do it in the computer because the matrices will be very big. So when you have a large molecule, say a protein with 3,000 atoms, then the matrix is going to be 9,000 rows and 9,000 columns. So it's quite a computationally intensive task to diagonalize this matrix, but it is possible. So that brings us to the end of this lecture. There's the recommended reading, which you should have a look at, and I will see you in the classroom session.